50 questions, not 5. Around 50. It'll probably be 45 or 50 questions. Multiple choice. All right. Then let's get into it. All right, so this is where we left off on Tuesday. We left off talking about proteins. We were talking about the monomer unit for proteins, which was what, again, I totally forgot. Someone please remind me. Monomer for proteins. Exactly. Anastasia, amino acids. That's correct. So the repeating unit molecule that we attach by covalent bonds for a protein is called an amino acid. So we string up all these amino acids together to get our larger protein polymer, which the fancy polymer word for a protein is a polypeptide or polypeptide chain of amino acids. And how do we build these proteins? How do we attach these amino acids together? How am I sticking them on, creating this chain of amino acids? What kind of reaction? Kelly, that's right. Dehydration synthesis. Yes, yes, totally. So when we remove that water molecule from the two monomers, we link them together with a covalent bond, dehydration synthesis reaction. Totally. And the opposite of which was when we added water and we lyse it, we lyse that bond, add the water, and it's hydrolysis. Hydrolysis is used to break monomers or polymers into monomer pieces, break apart these larger molecules into smaller ones. Okay, so that was a little quick review of last time. So today we're going to finish proteins and talk about our remaining 2 classes of biological molecules. So let's take a look at the slide here. To finish up proteins, I want to talk about protein structure. What proteins, oh boy, COVID, no, dry throat, I don't know. Okay, whew. I'm good. Proteins, structure of proteins, and why that's important, and what they look like. And so when we look at proteins, we have to look at, first, we know that we're building a long chain of these amino acids, right? And we looked at a few amino acids last time. We looked at what they look like, and we're attaching them together. And so we're building this line. We're building this sequence of amino acids. And it can be from a few hundred to a few thousand amino acids strung together, depending on how big and complex that protein is going to be. And so when we do this, what starts to emerge, what starts to happen, is that this long string of amino acids starts to fold on itself, starts to fold and create this intricate, beautiful, frickin' molecular thing. And it's a structure and it looks awesome. And we're gonna look at it right now. Okay. So what starts to emerge are these different levels of protein structure. The first one we call primary structure, then secondary structure, and this one is called tertiary structure, but we're going to write all this on the board. And so let's go ahead and do that. Let's define what I mean by these different levels of protein structure. What does it mean? What are we talking about when I talk about the primary structure of a protein? So protein structure primary. The primary structure, what I refer to when I talk about a protein's primary structure, it's the sequence of amino acids, the linear sequence, line. It's a line of amino acids. That's the primary structure. So it's this linear sequence of amino acids. So no folding or anything has taken place yet. Screen's blurry. Uh-oh. Is it? Is anyone else's screen blurry? Better now? Okay. Yeah, it's been doing that. Again, adapting. Good now? Okay. So I'll just kind of repeat what I said. The primary structure is this linear sequence of amino acids. It's this chain of amino acids, and it looks exactly like this. So here's our primary structure for this protein. And here are the amino acid subunits. These are all these different amino acids. Remember, there's 20 different amino acids. So we have glycine, proline, threonine, another glycine, threonine, glycine, glutamine, serine, blah, blah, blah. And they're all attached to each other in this sort of linear sequence. That's what we mean by primary structure. Secondary structure. I'm talking about secondary structure. This is when our protein begins to fold. So the protein starts to fold. And what we begin to see as a result of this protein folding are these shapes. When that linear sequence of amino acids, this is the linear sequence, starts to fold onto itself. Let me see. Okay, here it is. And it starts to fold onto itself. It actually starts to make these interesting shapes. And they're these coils that we call alpha helices, or an alpha helix. Kind of looks like that. We call this an alpha helix. And then we also start to see these sheets, these long sheets that kind of go on top of each other like this. And we call those beta pleated sheets. So I'll just do like, we draw that, they kind of look like arrows on top of each other. And these are our beta pleated sheets. And these folded shapes, we start to see that when we talk about the secondary structure of proteins. Okay. The last structure level we're going to talk about, there's actually 4 levels, but for this class, we're just going to talk about 3. Don't need it. You can talk about the 4th one in upper division courses. It's not like crucial at this time. But the next level of protein structure is called tertiary structure. It's our 3rd level of protein structure, tertiary. And when we talk about tertiary structure, we're talking about the final 3-dimensional form. Final 3D form. It's when the protein has completely folded, completely taken its final shape. Final 3D form. And if I said there's a 4th structure, you're probably like, how is that the final form if there's a 4th structure? That's a good question. Well, the 4th structure, it's actually when multiple protein subunits come together, like multiple tertiary forms come together. They do glob together, that's right. But don't worry about that. If we have this linear sequence of amino acids, its final folded form is its tertiary structure. And tertiary structure looks really cool. Let's take a look at it. Here we go. So by the way, here's our secondary structure. This is an alpha helix example. So when we start to see these, we're talking about secondary structure. And then this is actually, realistically, this is actually what proteins kind of look like. There are these alpha helices. You can see some beta sheets in here. And they look kind of like confetti. They're kind of neat looking. And they all look very different in the sense that they have different shapes and therefore different functions. Back to that main theme in biology, shape and function being very closely tied together. But they all have these sorts of loops and coils and sheets and pretty cool looking. Okay. What else did I want to say? Couple interesting points I wanted to add before we move on to lipids. The first one I'll say is that, like I said, there's, I want to say 20,000 different types of proteins, different types of enzymes, structural proteins, defensive proteins, proteins involved in movement, structure, all these things in the human body, okay, in our cells, in all of our cells. So if there's that many, the difference between all of these different proteins is the sequence of amino acids in their primary structure is different. So remember, 20 different amino acids, and you can make proteins from up to 100 or a few hundred amino acids to a few thousand. And depending on the order of these amino acids that you choose and the length, it's going to come out to a different shape, actually. So the primary structure and the specific order and arrangement of these amino acids, that actually determines the final shape of your protein. Yeah. And interestingly enough, as we said, the final shape is going to determine the function, right? So it all kind of comes back to that primary structure. And here you go. The function of that protein, again, whether it's an antibody fighting infection or a protein involved in muscle contraction, it's going to have a different 3 shape, right? It's going to have a different tertiary structure because it has the very different functions of these proteins, right? That we talked about last time. And again, what determines that shape is that order and arrangement of the primary structure, how long it is and which amino acids we chose in what order. And obviously, right, there's so many different possibilities. Some crazy possibility number because you got, you know, let's say in 100 amino acids, 20 different options for each amino acid. It's like, what is it? I'm not a math guy or stats, but it's like 100 to the 20th power, or 20 to the 100th, or something crazy. So a lot of cool options there. Anyways, okay. Questions about proteins before I move on? Proteins are awesome. Okay, let's move on to lipids. Lipids. Okay. All righty. Lipids. That's the first thing I wanted to put on our slide here. Hope I updated it on Canvas. I didn't used to have this bullet point, but there is no true monomer lipids. So if I didn't update it, go ahead and write that down. And I did mention that before. Lipids are sort of our oddball. They're the misfit of the group. They don't have a true monomer of repeating units. Lipids, as we'll see, are very diverse, and there's diverse looking and functions of lipids. So we're gonna look at that, but there's no true monomer for lipids. And really, because there's no true monomer for lipids, there's no true polymer. There's different looking giant lipid molecules, but they don't really have repeating units, right? And so we're going to take a look at one in particular right now that's very important, and it's the triglyceride. So let's write that down. Triglyceride. Tri, the prefix tri, like a tricycle has 3 wheels. That means 3. And a triglyceride is, well, first of all, the common word for a triglyceride is fat. Triglycerides are really when we think of things with high fat in them, foods like cheese or butter, there's a lot of these triglyceride molecules. And I want to show you what a triglyceride looks like. Here's a triglyceride. Tri meaning 3, right? And I'll show you why that is as well. First thing, a triglyceride has this, what we call a glycerol backbone. It's this glycerol molecule. Attached to it are 3 fatty acid chains. These are fatty acid chains. So take a look at that. You could also call them fatty acid tails. And you'll see that these fatty acid tails are just all these carbon atoms with covalent bonds between them and a ton of these hydrogens. We actually sometimes call them hydrocarbon chains. Hydrogens and carbons like crazy. And there's 3 of these guys here. 3 fatty acid tails attached to this glycerol. And that's our triglyceride. Okay. So let's talk about the function of lipids. It varies. There's a few important ones. Attached by what bond? So they're covalent bonds, all of them. All of these bonds are covalent bonds between all of these atoms in this molecule. Okay, cool. So lipids have a variety of functions depending on what lipid we're looking at. And so I'm going to write functions. Let's do that first. And a few examples. Triglyceride, especially, and fat, that mammals are really good at storing. The function for triglycerides and these fat molecules, and we store them in specialized cells called adipose cells. Adipose cells are filled with triglycerides for long-term energy storage. Okay, so let's write that. So we get a lot of energy from fats, and to store energy, to have an energy reserve of sorts, we store triglycerides. So that's one function, long-term energy storage. Very important cell structures are made of lipids. And we'll actually, at the beginning of next unit, we'll be looking at one in particular in detail called the cell membrane. It's made of a certain type of lipid. So cell structures, and in addition, certain hormones. Hormones are these chemical signaling molecules that are involved in development, growth and development, fight or flight response even is a lipid hormone called a steroid hormone, and the steroid hormone called cortisol, also known as adrenaline, is a lipid. That hormone's a lipid. Testosterone, estrogen. When we look at specific examples, I'll talk about that again. I believe that's coming up. Yeah, we'll do some examples of lipids, and I'll name those again, those steroid hormones. And last, another important function is heat, insulation. Heat, insulation. So lipids are important. They do some important things for us, for sure. And again, we're gonna see that steroid hormones, triglycerides, cell membranes, the structures of these lipid molecules look really different. There's no consistent monomer that we're talking about here. Okay. So I believe that's all I wanted to mention. Oh, we just did functions. Yay. Okay. Another important concept, saturated versus unsaturated fats. Okay. I'll just leave that up for a second while I erase. Okay, so here on the top, you can see what we call a saturated fatty acid, and below, an unsaturated fatty acid. So we're actually, part of the reason I showed you what a triglyceride looks like and what these fatty acid tails look like, is because you can have different types of fatty acid tails in a fat molecule or our triglyceride molecule. And so we're gonna look at these two different types. You can have a saturated fatty acid tail or an unsaturated fatty acid tail. We're gonna look at the differences between the two. And they're important for certain properties of lipids. So let's start with saturated, top one. So I'll write saturated, FA for short, fatty acid. So our saturated fatty acids compared to our unsaturated, there are more hydrogen atoms. It's actually why we call it saturated. It's saturated with hydrogens. So a few examples of saturated fats. Animal lard, butter, cheese has more saturated fat in it. And importantly, something to notice about lard and butter, besides tasting good, is that they're solid. They're solid objects, right? Not liquid. They're solid at room temperature. I'm gonna just do a little division here. I'm gonna write solid at room temperature here. And I want to tell you why that is. I'm going to draw a simplified version of a saturated fat. A triglyceride molecule with saturated fatty acid tails. Here's our glycerol backbone, and here are the fatty acids that come off of it. So I'm going to draw two more. Okay. See how they stack up really nicely? That the fatty acid tails are very straight, so they can just stack really well. I want you to keep that in the back of your mind, and then we're going to come back to that fact. Because we'll see with our unsaturated fatty acids that that is not the case. They do not stack really well. our unsaturated fatty acids have less hydrogen atoms. The reason this is, we didn't really go in depth on double bonds in our chemistry unit, but that's okay. You don't have to know really why there's less hydrogen atoms, but if you think about it, there's a double bond, they're sharing more electrons, and there's not going to be, to get to that 8 electron in its outer shell, it doesn't need an additional hydrogen. It has to do with that. But I want you to know, when we talk about unsaturated fatty acids, there's less hydrogen atoms, and this is because there's the presence of these double bonds. So I'm going to write contains, in the unsaturated fatty acid tails, there are double bonds. Contains double bonds. I'll just go ahead and show you what that looks like. So there are double bonds where my mouse cursor is. Instead of a single covalent bond, it's a double covalent bond. And check it out. See how this saturated fat, very straight? This one, the unsaturated fatty acid, not very straight, is it? It kind of has these bends and, little, well, we call them kinks in the molecule. And actually, it's because of the double bonds. The double bonds induce these kinks and these bends in the molecule, makes it go upward or down, just gives it an angle, right? It's not straight. So let's write that the double bonds produce kinks. In the molecule, or the fatty acid tail. So I'll write produce kinks in fatty acid tail. Okay, before I show you what it looks like at room temp, let's write some examples of unsaturated fats. These are healthier for you, and really oils, any like vegetable oils, etc. Oils are unsaturated. Their triglyceride molecules have unsaturated fatty acids. Sort of ran out of room here. So what I'm going to do, I have this written down by now. So I'm going to erase here. We're still talking about unsaturated fatty acids, but just write this underneath. And I'm going to compare it to the saturated fatty acids. When I draw out what these triglycerides look like. So here are the glycerol backbones. And then because of these kinks or these bends in the fatty acid tails, it's going to look like this. you think these are going to be able to stack up on each other really well? No. They're going to fall, they're going to slip, and because of this, they're constantly moving, and it makes a liquid at room temperature. All right. That's it for fatty acids. Let's go ahead and, so we've really defined and classified what lipids are, their function, a little bit of their structure here. As far as triglyceride go, let's go ahead and talk about some examples of lipids and some important examples of lipids. Examples. Well, we talked about a few already, right? Just with oils and butter, right? So let me stop that. So oils, butter. Some examples of triglyceride lipids, right? And I mentioned this when I talk about cell structures. Cell membrane. Cell membrane is made of a special type of lipid called a phospholipid. And the cell membrane, when we look at cells in the next unit, is this barrier, this protective barrier that separates the inside of the cell versus the outside. And it's made of a type of lipid. So clearly very, very important. Mentioned it, steroid hormones. This includes growth hormone, human growth hormone, hormones in development, developmental. So that would be like testosterone, estrogen. These are steroid hormones, and they're considered lipids. And interestingly enough, why I say there's no really true monomer, polymer, I mean, steroid hormones look like this. And the next thing we look at looks very similar to this. It's like this ring structure. That's kind of like what a steroid hormone looks like. These rings attached to each other. Clearly very different than our triglyceride, right? Last important example of a lipid that plays a few important roles in our cells is cholesterol. But the one that the role that we're going to focus on mainly in the next unit, but there might be a test question on it for this exam. I forget. I might have included it, but to be safe, know this. Cholesterol stabilizes cell membranes. So you find them embedded in the cell membranes that surround our cells, which are also, right, made of these phospholipids. So you'll find cholesterol molecules. Just in case I did include a question on it on the exam, I'll just underline it in red. Cholesterol actually looks a lot like this too. In fact, you synthesize and create testosterone and estrogen from cholesterol, interestingly enough, but that's just a fun side note. All right, so I'm ready to move on from lipids, but are there any questions so far? Lipids, etc. Before we move on to our last class of biological molecules. We've done carbohydrates, we've done proteins, we've done lipids. We have one more to cover for today. All righty, let's do it. It's our nucleic acids. So this is our fourth category, nucleic acids. Not to be confused with amino acids, right? It's our fourth category of biological molecules, nucleic acids. All right, so we're back to this sort of rule where there's a defined monomer, a repeating unit that will form a polymer. And the monomer for nucleic acids is called a nucleotide. It's a nucleotide. We're going to look at the structure of a nucleotide. We're going to look at, actually more specifically, we're going to look at the structure of a nucleotide that we would find in a polymer, for a nucleic acid polymer that I'm sure many of you have heard of, called DNA. DNA. So DNA stands for deoxyribonucleic acid, DNA. Or as my brother says, drugs and alcohol. Ha ha ha. Just kidding. No. Deoxyribonucleic acid. So we're talking about nucleic acids, right? And this deoxyribo part, we're gonna look at why DNA is called deoxyribonucleic acid. And we're gonna look at why this is by looking at what a nucleotide that we'd find in a DNA molecule looks like. So every nucleotide that we see has 3 parts. So 3 parts. So our nucleotide has a phosphate group, phosphate group is this chemical functional group that consists of a phosphorus atom and 4 oxygen atoms, PO4. And importantly, it has a negative charge associated with it. Sometimes even a -2 charge. So I'm going to put PO4 2-. It's very important to know that this phosphate group carries a negative charge. It's going to confer different properties to nucleic acids. Okay, so we have a phosphate group attached to which we have a sugar group. And in the case of DNA, the nucleotides of DNA, which is actually a polymer, it's an example of a polymer nucleic acid that's many nucleotides attached to each other. The sugars in the nucleotides of DNA are deoxyribose. And that's where the deoxyribo comes from. It's just the specific sugar, type of sugar we find in the nucleotides of DNA. And don't worry, we're going to look at other polymers as well in the next slide. DNA is one of them. And the last part of our nucleotide, third component here, is the base, or nitrogenous base. Nitrogenous, because it's nitrogen containing, it has a lot of nitrogen atoms in the base. Nitrogenous base. And we're using DNA as an example here. Because I want to show you that there's actually 4 different nitrogenous bases that are possible in a nucleotide of DNA. Other polymers, nucleic acid polymers like RNA, your options are slightly different for your nitrogenous base, but for DNA, that's a good one to start with. You have a deoxyribose sugar in your nucleotides, and you have 4 options for your nitrogenous base. You choose one for each nucleotide. And our 4, 2, 3, 4. So I'm going to write maybe I'll just write like 4 options. Move that out of the way. This is our PO4. So I'm going to write 4 options for your base here. You can have an adenine, a thymine, a cytosine, or a guanine. In your nucleotide for DNA. So interestingly enough, the DNA for humans is about 3 billion nucleotides attached to one another. So that's pretty interesting. So a lot of nucleotides for the DNA within human cells. Oh, thank you, Sophia. Yeah, guanine. These are 4 nitrogenous bases that we can choose from. We choose 1 for each nucleotide. And you'll see actually that we abbreviate our bases. Like if we're looking at a sequence of DNA, we're looking at a lot of nucleotides, right? So we'll often abbreviate adenine A, thymine T, C, and G, cytosine and guanine. These are our 4 bases. So let's take a look at a nucleotide, just kind of what it actually looks like here. So this is it in its chemical structure form. Here's our phosphate, and it has that minus charge, right? Here's our sugar, and then here's our nitrogenous base. In this case, it's an adenine base. This is what adenine looks like. The other bases look similar, but all of them have nitrogens in them. How are we doing on time? I haven't even looked. It's like we're an hour over. No, we're good. All right. Might even end early. Okay, so let's talk about some examples. We already talked a little about DNA. We didn't really discuss what DNA does, what its function is. But let's look at some other examples of nucleic acids. Uh-oh, what cut out? Something cut out. Little spotty. Well, isn't that great? Well, I'll repeat what I said. I basically said well, the last thing I said, well, I just showed you this chemical structure of a nucleotide. The phosphate with its minus charge, our sugar here, and nitrogenous base. And this one happens to be adenine. Did you guys get that? And then I said we're going to go over some examples of nucleic acids. Okay, cool. Okie dokie. So examples and functions. Examples of different polymers of nucleic acids and their functions in the cell. Well, DNA, let's start with that, right? Fancy word, it's not even fancy really, for polymer of a nucleic acid is polynucleotide. So many nucleotides strung together. The function of DNA is to store genetic information. And yeah, that's really it. Store genetic information. And for the next few, I want you to just keep in mind, this lecture is focused on at the surface level of these biological molecules. Throughout the semester, we're going to explore these functions a lot more in depth. So it might not make sense right now, but that's okay. I just want you to know it as a foundation and for your exam. But this is a foundation, and then later on, we're really going to explore what RNA and ATP do in detail. But I do like to include other examples here of nucleic acids. RNA is involved in the transfer of genetic information. And even protein production. You have a whole lecture devoted to that, so just being aware of it now is what I want. And having an idea, knowing that RNA is involved in transferring this genetic information and producing proteins. It has a role there. And ATP is actually the energy currency of the cell. We explore that a lot in the next unit, actually. Whenever there's a chemical reaction or a process that requires energy, the cell will use an ATP molecule. It's a high energy molecule. You can think of it as like a battery. And it's a nucleic acid. So because you have an exam, there's actually a fun little, not exactly ending early. There's 2 minutes left. I have a fun little activity that'll help you practice some of this. In the way, you're probably still writing. But I have a fun little activity I want to do on the next slide, just answer some questions. Of course, you're not turning this in or anything, and it's just for fun. But let's determine if these are true or false. And for any false, let's explain why, and then we'll call it a day. DNA is a polymer of amino acids. True or false? False. False. Let's correct it. Why is it false? DNA is a polymer of what? Nucleotides, absolutely. And amino acids are the monomer for proteins. You bet. Good job. Glycogen, starch, and cellulose are all monosaccharides. True or false, and why? Oops. Correct, Anastasia, that's right. So false, they are polysaccharides. Great, it's 4:15, so let's just race through these. And it's recorded, so you can review it. Saturated fats are solid at room temperature because the double bonded carbons in their fatty acid tails. True or false, why? What do you got to replace there, if anything? That's correct. It is false. They're solid at room temperature. They have more hydrogens. There's no double bonds. Absolutely. That's correct. Okay. The final three-dimensional shape is determined by its primary structure of a protein. True or false? Yes, true. Nice. A dehydration synthesis reaction would combine amino acids to form a protein. Perfect. Awesome. All right, well, that's it then. So I'll email out those announcements again, and that's it. So I'll see you, I won't be seeing you Tuesday. I'll be emailing out a recorded mini lecture for the lab with lab materials. Good luck on the exam. Next time we meet, lab due tomorrow, so I'll be creating that assignment on Canvas for upload. Yes. Oh, if you have questions about the lab, yeah, I can stick around if that's what you're referring to. But yeah, otherwise, that's it. Lab quiz? No lab quiz, just the Canvas lecture quiz. There's no lab quiz, there's only a lab exam in a few weeks, I believe. The 25th? No, it's not that soon. Of October, maybe. We still have a few labs to do before the lab exam. 13th of October, thank you. Yeah, so that's, yeah, about 3 more weeks. Just the lecture quiz tomorrow and then the lab or lecture exam Tuesday. Yeah, so does anyone have questions about the lab or the lecture today or anything? Because I have office hours right now, so I can totally answer anything. Really? All right. Well, maybe you're typing. I'll just sit for another minute here. Out of water. Okay, well, if anything at all comes up, then email me, of course. Is there a quiz on the biomolecule lab? No, that it'll just be included in the lab exam. That's on the 13th. Yeah, the quiz is on lecture material. So, and I believe it's biological molecules. So there won't be any lab specific questions. It'll just be about things like this, things we've talked about in lecture. Yeah. For lab, the only testing component are the lab exams. And there's 2, I believe. You're welcome. All right, guys. Well, that's it. So have a great weekend. I'll see you all Thursday. So in a week. Good luck on the exam. Bye, everyone. And I'll make the assignment on Canvas so you can upload your labs. I'll do that now.